Hello, this is Dr. John Lewis. I'm going to be talking to you today about the importance of polysaccharides for brain health, immune function, and overall quality of life. I am the founder and president of Dr. Lewis Nutrition. I am a voluntary associate professor at the University of Miami School of Medicine in the Department of Psychiatry and Behavioral Sciences, and I'm also an advisor, diplomat, and faculty member of the Medical Wellness Association. My contact information is also included at the bottom of this slide. I would like to acknowledge two people in particular who have had a big impact on my life, and those people would be Dr. Reg McDaniel, a pathologist from the Dallas area, and Miss Barbara Kimley, a patient at the University of Miami, both of whom introduced me to the importance of polysaccharides. Dr. McDaniel from the aloe vera side, and Miss Kimley from the bio-brand side, the rice brand side, excuse me, and I owe them deeply for what they introduced to me because I learned about as much about polysaccharides as probably fit in one lecture, and most of what I learned in grad school was about the benefit of polysaccharides as an energy source, not having anything else to do with their immunomodulatory or other capabilities. So I'm always indebted to them for basically changing the course of my life. And I'm also indebted to the sponsors and the subjects who participated in our research over the years. Without both of those entities, we never would have been able to publish anything today. I want to start off with just a very simple quote that probably most people have heard, and it was said by Hippocrates, the father of modern medicine. He said this many, many years ago, way before the Western calendar even started, and that is, let food be thy medicine. I think that is such an important quote to always consider as we not only think about polysaccharides, but not even just nutrition, but actually nutrition in the context of where we are today, not only as a country, as a society, even as a species, in terms of all of the chronic diseases that we are riddled with. And quite frankly, the majority of these chronic diseases are rooted in the fact that we do not eat well anymore. And so food is such a powerful component to our health. In fact, in my view, it's the most important component. And we need to always keep that in mind as we're thinking about how health is. It should be actually our number one priority in life. Unfortunately, for many people, it is not. But we will address one aspect of this in this lecture today in terms of the benefit that polysaccharides have and how truly polysaccharides are part of that concept of letting food be thy medicine. James Lind, Dr. James Lind, is someone who is not as famous as Hippocrates, of course, but he's a person who made a very significant discovery for the British back in the 1700s. You can see from the slide here the dates that he was alive. Dr. Lind actually figured out that the use of citrus could prevent scurvy. And with Dr. Lind's discovery, what happened beyond not only allowing the British to dominate the world for many, many years, but probably also is the reason why even we speak English, the English language in the United States today, is due to this gentleman's discovery, where he discovered at that point in history, in man's history, people were dying of scurvy when they would leave Europe, set sail for the Americas. Many people would die of scurvy. And so with his discovery of using lemons, limes, and oranges, he could actually reverse scurvy and prevent it from happening in the first place. So when you think of citrus in the context of preventative health, if you will, or even reversing a health condition like scurvy, that's very powerful. So I'm quoting Hippocrates and the discovery by James Lind to tell you that the concept of nutrition, the importance of nutrition, is a very old concept. This is nothing I came up with. This is nothing even recent. This has been around for a very long time, and it's important for us to always keep this in mind once again as we think about our health and how the context of health should really be completely changed from what it is today. The flip side of that, of course, is the shape that we find ourselves in today in terms of why we're where we are today. So I will mention these epidemics in a moment, but really when you think about where we are today, it's due to the standard American diet. over-reliance on too many things: processed food, animal food, sugary food, fatty food, people eating too much of this and not enough of that. So too much processed fried food, not enough whole food, plant-based food, too much animal food, not enough raw fruits and vegetables. So really, when you think about it today, the average person, it's not just an American problem, it's a worldwide problem. The average person today is overfed and undernourished. We have a tremendous issue with our food today in terms of not only the way we look at it, but the way we utilize it for our health. So really, really huge. In my opinion, it's the biggest problem today. So feeding into that, no pun intended, we have epidemics of all of these different chronic diseases. We have obesity is running rampant. We have cardiovascular disease, still the #1 killer, although cancer, it may be #1. It just depends on the data. Type 2 diabetes is out of control. Osteoporosis, arthritis, chronic pain, mood disorder, on and on and on. All of these things have direct links to our behavior and specifically to our nutrition. Of course, exercise and physical activity are very important as well, but in terms of the science, what the science shows, nutrition is without question our #1 modifiable lifestyle factor when it comes to either preventing chronic diseases in the first place, or when we get something like this, being able to reverse it. I mentioned obesity. Obesity is completely out of control in the United States today. Look at these data. These data show very clearly that nutrition is the driving factor here, and I will get to that in just one moment. But when you look at these data, in the early 1960s, about 10% of men and about 15% of women were considered obese. This is defined by a body mass index of 30 or greater. In less than 50 years, look what happened to us. Now we have actually more men who are obese for the first time than women. We have about 43% of men and about 42% of women who are now considered obese. That all happened in about a 50-year period. Our genes did not evolve this way. Our genes just didn't simply say, okay, it's time for humans to be fat and get sick. What happened in the late 1950s, early 1960s? The advent of fast food. What else happened? We had the Green Revolution in the 1960s. These things have been significant drivers in terms of now why so many people are not only obese. There's no such thing as being fat and happy. That's a myth. You not only have obesity, you have heart disease, diabetes, cancer, mood disorder, on and on and on, chronic pain. All of these other things go along with this. So we have significant issues today as a population, as a country, as a society, where so many people are now sick. There's no better way to say it. We have to get a grip on our health. It's personal responsibility. It comes down to what each of us do every single day. And that's really what is healthcare. It's not some nebulous term that the government uses that by the way, we're now spending $4 trillion per year throwing money into, and we still cannot get a grip on all of these different chronic diseases. But this slide very clearly shows the root of the problem, and that is driven by this modified diet that we've gone from in the early 1900s, where most people were somehow involved in agriculture, to today, less than 1% of us is involved in agriculture, and it's all shifted. It's all about the food that we're consuming and what we are doing to ourselves. So what about dietary supplements? I've kind of given you this sort of broad overview of where we are as a society, but what about dietary supplements? Unfortunately, typically, the person who is ignorant of dietary supplements in general, they have this misnomer that, well, there are, inconsistent findings, or they're not FDA regulated. That's a common comment that I get from people. They think that, oh, well, if it's not regulated by the FDA, then there's something wrong with it. That could not be further from the truth, but that's another topic of conversation that's not really germane to this lecture. Nonetheless, there are about 80,000 dietary supplements on the market today in the United States, and the Institute of Medicine suggests that there are about 1,000 new products that come online every single year. So regardless of what maybe some skeptic thinks, it's a huge industry. Here you can see the value of the industry, starting with the 1994 passage of the DSHEA Act all the way up until just this last couple of years, you can see that the industry has been steadily growing pretty much every single year, regardless of whether there have been recessions or corrections in the overall economy. The dietary supplement industry continues growing. So what this also tells us is that people are looking for alternative answers. People go to their conventional medical provider and they're still sick. They're taking medications. They're taking more medications to treat the symptoms of the first medications. People want answers. People want solutions. People don't want to continue taking medications. So finally, we have people turning to nutrition or dietary supplements to help them with whatever it is that they're dealing with. And again, we have lots of problems today. Just quickly, the typical dietary supplement use data. This is from the CDC from within the last couple of years, if you think the CDC still has much credibility at this point. But nonetheless, about 58% of Americans who were 20 years of age or older had used at least one dietary supplement in the last 30 days. And then the greatest overall use of dietary supplements was about 80% of women 60 years of age and older. So dietary supplement use is increasing across the board, basically among both sexes and across all different age groups. The typical dietary supplement user in the United States is an older, non-Hispanic white female who is physically active and has an above average level of education. So again, you can see from a perspective across the population, we have lots and lots of Americans who are using dietary supplements, and I'm glad to hear that. I'm glad that that trend is moving in that direction. So now let's get into some of the specific findings that we conducted here at the University of Miami Miller School of Medicine. We have spent many years looking at a couple of different classes of polysaccharides, and I'll get into those specifics as we move forward. But we've spent many years looking at the effects of these polysaccharides in humans. We've been conducting clinical trials, meaning that these are studies in human beings. We're not looking at studies in mice or rats or tissues or cells. We are looking at how these polysaccharides affect actual people. So the results are directly translatable. You don't have to worry about thinking, oh, well, this was mice research or this was only effective in a cell or something. No, this was looking at the effect of these polysaccharides in actual people. So I'm happy to discuss with you the findings of our research program, and we're still hoping to continue that line of research as we move forward. So you may be thinking at this point, well, what is a polysaccharide and where do they come from? So polysaccharides are complex simple sugars or complex simple saccharides. Saccharide is a term meaning sugar. I could have said, well, sugars are good for you. You would have probably turned off the lecture at this point, but actually it depends on the type of sugar. So simple sugars like glucose or fructose may not necessarily be the best thing to consume in large quantities. In fact, I'd probably say based on the literature, they're not. However, complex sugars, polysaccharides, are a whole different animal, a whole different kind of biochemical input or information for our cells. And unfortunately, because of the way we've changed our food, as I mentioned a few moments ago, we now no longer get these polysaccharides in the modern diet today. So it's important actually to get these polysaccharides from food sources that are concentrated down into a dietary supplement. That way you are getting a very concentrated dose and you're not skimping on the benefits that these polysaccharides can give to literally every cell in your body. So specifically to our work, polysaccharides come from some of these plants, rice bran, aloe vera, diascorea, or wild yam. These are definitely plants that provide significant amounts of these polysaccharides in doses that can be very beneficial to us. So the first segment of the talk, the first group of research results that I'll talk about is from a product that we generically termed allopolysaccharide multinutrient complex in two different studies, one with adults with relapse-remitting multiple sclerosis and another in adults with moderate to severe Alzheimer's disease. So these are obviously two of the three main neurodegenerative diseases, the third one being Parkinson's, of course. And when you look at all of these different neurodegenerative diseases from a conventional medical perspective, basically there is nothing to offer these folks. If you can maybe keep the person from declining, keep that person stable, that may happen for a while, but in general, people continue declining, they continue falling off the cliff. So in terms of what we know about these diseases, why they are caused, it's also very unknown at this point. But then what do you do for these people once they have one of these conditions? That's really the key. And so we wanted to look at whether or not nutrition, and in particular, nutrition that included a significant concentration of these polysaccharides from aloe, could actually make a difference in these people's lives. So another thing that you may be thinking of at this point is, well, why are these polysaccharides so important, and why do our cells need them? So this is a graphic representation of basically every cell in the body. And every time the cell does something, it has organelles or tiny little components in each cell. Organelle is like it's a representation of an organ in our entire body, but in each cell we have what are called organelles. For example, the mitochondria, our engine, the endoplasmic reticulum, the Golgi. So these are different parts of the cell that are called organelles. And the organelles take all of this different information that we primarily give it through the diet, and then it constructs them into different compounds or molecules that it needs to function properly. So broadly speaking, this is called the bioengineering of life. So whether these organelles are taking things, for example, like oxygen, vitamins, minerals, other elements, amino acids, fatty acids, polysaccharides, it's constructing these different nutrients and components into other cells that we need to function properly. Or in many cases, it's replicating itself. Cells go through this process of replicating themselves. So every time the cell does something, whether it's constructing another cell, whether it's talking to another cell, whether it's creating a bioactive compound that then tells another cell to do a job, these are all synthetic activities, metabolic, synthetic, cellular activities that our cells do every single moment of their lives, and we constantly need to give them the nutrients or the raw materials that they need to function properly. So if you think about, if you've had statistics, you know there's a concept called garbage in, garbage out, meaning that if you run a study and you collect poor data, the best statistics in the world cannot make your study into something that it was not. Same thing with our cells. We need to give our cells the proper raw materials that they need to function properly. And the best way that we can do that is through our nutrition, through the things that we put into our mouths, whether we eat it or drink it. So all of these things are very much crucial to our health, because we have approximately 30 trillion cells in our body, and all of those cells work together to form organs, to form organ systems, and then to form us. And so every single cell is important, and that is why these polysaccharides are so crucial, because of this step in between the endoplasmic reticulum and the Golgi. These polysaccharides are needed, and in particular, they're needed in certain ratios. And when we give our cells those polysaccharides that they need, man, now the real bioengineering happens, and the cellular activity happens to make us function properly, and that's why they're so important. So one of the things that we think about in terms of, again, the ultimate effects, things that we can see on a clinical or conscious level of why these polysaccharides are so crucial, is that they seem to be signaling mechanisms for our immune system. And as you know, our immune system is so important. Our immune system overall is our defense mechanism, right? It's from keeping us, it surveys for, infectious agents. When we have trauma, it sends out signals to inflame that area, to repair it. The immune system talks to all of our other major organ systems. It's so crucial to have a healthy, functioning immune system. And so it appears like these polysaccharides are very important for keeping our immune system functional and surveillant. Here is a patient that actually Dr. McDaniel was working with, who, around the time that I met Dr. McDaniel, he was telling me about all of the amazing discoveries that he had had in his career, and one of them happened to be with this particular cancer patient who had been put into hospice and was basically given a terminal prognosis. Now, this person had had surgery, chemotherapy, radiation, everything, and unfortunately was still sent to hospice. So you can see in an image here of the liver where this tumor is very big, where it's very, very defined. And after about a year later of taking the polysaccharide, you can see the reduction in the size of that tumor. And then ultimately, this person ended up leaving hospice cancer-free. So this was one of these initial learning moments for me where I was, as rapidly as I could trying to learn and really understand why these polysaccharides are so important to us. But this particular person's case was just so profound to me. And there have been so many others as I've gone through this journey over the last going on 20 years that it's just amazing to me how time and time again these polysaccharides continue to demonstrate how effective they are. So just real quick, here are a few very, very sad statistics about dementia and Alzheimer's disease in general. There are approximately 44 million people worldwide suffering from Alzheimer's today. Of those 44 million, about 6 million are Americans, and we have another 8 or so million people with different forms of dementia in the United States. It's the leading cause of institutionalization among the elderly. It's the 6th leading killer in the United States. It's actually the #1 leading killer in the UK, which is just mind-blowing. Out of all of the top 10 killers, it has no conventionally thought of treatment, cure, or management. Now think about that for a second. With all of the things that conventional medicine attempts to do for people with various forms of heart disease, cancer, or diabetes, when it comes to comparing those treatments or those attempts to help people with people that have dementia or Alzheimer's disease, it's completely night and day. And then when you think about these costs, I mean, the costs are just outrageous. We're talking about just in the United States for 6 million people with Alzheimer's, roughly $500 billion in formal and informal care costs. Those numbers are just staggering. It is really unreal what this country is spending, not only in terms of actual money, but in terms of the lost quality of life, not only for the individual who gets dementia or Alzheimer's, but for the caregiver who's the most affected person. And actually, caregivers of Alzheimer's patients are the ones who suffer the worst compared to any other type of caregiver. And then all those other people around that dyad of the patient and the caregiver, all those other secondary caregivers and everyone else. It's an incredibly sad disease that, again, we have virtually no answer for at this point. There is no scientific consensus on what causes dementia, let alone how to treat it. So it's an incredibly sad disease, and pretty much everyone knows someone today, either a direct family member or a close friend who has had the disease and succumbed to it or currently has it today. It's just a tragic, tragic disease. So as I mentioned, we ran a trial in people with moderate to severe Alzheimer's, which, by the way, Big Pharma doesn't really care anything about studying those people. Big Pharma wants to study either newly diagnosed people, very early onset, or people who only have a mild severity of the disease. They're not really interested in studying people that have progressed beyond that because they look at those folks as not being worthy of study. So we chose to look at the sickest of the sick, and we looked at these people over a 12-month period. We didn't care what medications they were taking. We didn't care what other conditions they had. We wanted to look at the effect of this formula of the allopolysaccharide combined with some other different nutrients that we thought were important to see how this formula would impact cognitive function, immune function, and inflammation. We looked at blood work at baseline in 12 months, and then we did the neuropsych testing at baseline 3, 6, 9, and 12 months. We gave people 4 scoops of the powder per day, which is a scoop is roughly 2.5 grams. So we felt like that was a significant or enough of a dose to show some benefit at the end of the study. So perhaps the most exciting, among many different exciting things that we showed in this particular study, perhaps the most exciting finding that we found was a clinically and statistically significant improvement in cognitive functioning in these folks according to the ADAS-Cog. The ADAS cognition score is widely considered to be the gold standard in terms of assessing dementia, I'm sorry, assessing cognitive function in people with dementia. So the fact that we actually showed something clinically and statistically significant was unheard of. We published this first paper in 2013. Here we are 9 years later. We still have not seen anything else in the literature, either before, during, or since our work, that showed anything close to what this showed. So we were incredibly excited by the findings of our study on that level. Again, to have people actually start remembering things, start recalling things, being able to have focus and alertness, and to be able to demonstrate, hey, I'm back, I'm a person again. And so that was really, really exciting to us. In terms of some of the biomarkers representing immune function inflammation, we showed a really nice finding related to the CD4 to CD8 ratio, which is an overall indication of immune function. We want that ratio to be as high as possible, and not just within the context of Alzheimer's, but actually within the context of just getting older. That CD4 to CD8 ratio needs to be as high as possible, and so we were incredibly happy with that finding. TNF alpha and VEGF both have been classically looked at in heart disease and cancer. Definitely if they're dysregulated, if they're too high, then that's not a good thing. That means that you have definite chronic inflammation. And so to be able to lower that inflammation according to both of those markers was a very nice finding. And then also the CD14 cells, which are a marker of adult stem cells, we showed just under a 300% increase in those cells. So we were just incredibly excited about all of these findings, and it really made our work, very rewarding on that level. Let me just talk about, let me go back. I'll keep going here. We did a secondary analysis. We looked at a protein called brain-derived neurotropic factor. When we ran the study and then published that first paper in 2013, there were other papers that were being published related to brain-derived neurotropic factor, or BDNF, and we had our samples stored in a freezer, so we wanted to look at whether or not the allopolysaccharide complex actually had a positive impact on BDNF. So we ended up running the labs on that. We did that analysis, and then we did a breakdown of 5,000 units where we had looked at BDNF in a previous study in people with HIV, and we found that there was a significant change in terms of people that had more than 5,000 units of BDNF versus people who were under that 5,000 marker. So we were able to use that as a cutoff to then make comparisons related to cognition and immune function. So we actually published 2 other papers once we did that secondary analysis looking at BDNF. As I mentioned here, BDNF, again, as we were publishing our first paper, and then in the subsequent years after that, other papers started showing the real importance of this protein. So BDNF is very critical for this neurosynaptic process in terms of plasticity, potentiation, learning memory, higher order thinking. All of these things were being published, and so we felt like, okay, well, let's see, again, within the context, maybe the first time of looking at what's going on in people with Alzheimer's in response to a dietary supplement, could there be some benefit to that, and would it have an impact on BDNF? And then could we get maybe a bit more of a mechanistic understanding in terms of all of the clinical results that we showed in the first paper? Could the findings with BDNF actually help us to show even more mechanistic, I guess you could say, evaluation or explanation? So again, we published 2 more papers from this study. Unfortunately, we didn't show a statistically significant change in BDNF. It did go up, and then the pro-BDNF, which is the molecule that is the precursor to actual BDNF, it basically stayed the same. But we showed about an 11% increase in BDNF over the 12-month period. So it didn't change statistically, but nonetheless, it is going in the right direction. And maybe if we had had a larger sample size, we would have showed something statistically significant. So the first paper was related to cognitive function, and you can see here again, by splitting the subjects into those who were at 5,000 picograms or above versus those who were under 5,000, you can see a very clear delineation. All of these different cognitive function scores were actually better in the people who had a higher BDNF level. So there's definitely something to that idea that if your BDNF level is above 5,000, then you're going to have better cognitive function. So that was really a nice finding for us. Although again, there's a bit of mystery behind it in terms of what actually is going on here. We didn't have the funding to do imaging studies in this particular study, so it would have been nice had we had that data. But nonetheless, we do show some really nice findings here. Same thing with immune function and inflammation. Again, you're finding or you're looking at overall a better immune function, lower inflammation, if you will, by people who have this BDNF value over 5,000. So again, it was interesting from a mechanistic perspective to say that some of these findings definitely seem to have contribution to BDNF. It's hard to say exactly how much contribution BDNF is making here, but clearly we know that BDNF is playing a role in the fact that we were actually able to show, even though it was non-statistically significant, at least an 11% increase in BDNF at the end of the study. It would have been interesting too if we had had more funding to draw blood at the 3, 6, and 9 month follow-up points. And again, unfortunately, we didn't. So we were limited to only drawing blood at baseline and 12 months. So it would have been very interesting to have looked at what else was going on, during the study, in the middle of the study. But unfortunately, we didn't do that. But hopefully, we can in future research. So just to sum all of that up, for the first time, and still even today, after nearly a decade later, I don't know that anyone has replicated our work. We showed clinically and statistically significant improvements in cognition. We showed improvements in overall inflammatory status, which we theorize links to neuroinflammation, which is obviously very important. We showed an overall improvement in global immune function, and then this ability to really turn on the adult stem cell making process. And remember, these are people, I didn't mention this before, but on average were 79.9 years of age, had been diagnosed with Alzheimer's at least for 1 year, and on average had it for over 3 years. So these were very impaired people. And by the way, not only were they that old and had that serious of a disease, they also had other things. They had heart disease, they had depression, they had diabetes. So these were really, really advanced cases, and we were still able to show very, very significant findings in our first trial in people with Alzheimer's. The second set of studies I'll talk about is within the folks having relapsing remitting multiple sclerosis. These were also very impaired people that had the disease for on average over 12 years. We used the exact same format in terms of between the Alzheimer's and the MS study. We looked at blood work at baseline and 12 months, and then we did not exactly neuropsych testing, but we did more functioning, and quality of life measures, because the cognition issue or the cognition story is not exactly the same for people with MS as it is for people with dementia and Alzheimer's. For people with MS, it's much more about functioning and how the disease affects their ability to really live and their overall quality of life. But we did look at a lot of the same different group of inflammation markers and immune function markers. They were slightly different, but for the most part, they were pretty much the same. So we were able to publish 2 papers from this article. We looked at, in the first paper, the infections, which, by the way, infections are the leading killer of people with MS. They're also significantly what's driving the cost of MS. People with MS obviously have an impaired immune system, and they're much more susceptible to different types of infections. So not only does that kill people with MS, the leading cause of mortality, but it's also driving a lot of the expenses around MS treatment. And then the second paper was related to, again, the functioning quality of life measures that we looked at. So let's jump into these findings. Again, infections is the leading killer of people with MS. So look at these numbers here. At baseline, you had just under an average of 8 infections in the folks with MS. 12 months later, that rate went all the way down to 2.5. Incredible finding. I mean, again, nothing like pretty much anything in the literature in response to taking this allopolysaccharide-based complex and then seeing what happens in people with this type of disease. So again, we know from previous research that the allopolysaccharides are highly antibacterial, highly antiviral, and the fact that they were able to knock down these occurrence of infections so significantly, so statistically significantly, was just really exciting to us. We had split them up, as you can see, in a few different types of infections, but really focusing in on the total number, which is the most important, going from 8 to down to 2.5 was just really, really powerful for us. And then looking at the inflammation and immune function markers, we go into a lot of detail related to some of the findings here, which if you look at IL-2, you may have thought that it should have actually gone down instead of increasing. But we explain that quite a bit in terms of some of the other things going on here, especially related to IL-1 beta and EGF. So it actually is not difficult to understand when you look at the results in context of why people with MS have all of these infections, and then the way the polysaccharides are affecting the incidence or occurrence of those infections, and then how that relates to some of these markers in the immune system. But definitely, for example, the CD95 or the FAS marker is kind of like CD4 to CD8 ratio. It's an overall marker of immune function, and that went up very highly. So we actually show something here, which is, in our opinion, immunomodulation, meaning that the body is reacting to the materials that we're giving it, particularly these allopolysaccharides, and it's responding accordingly. And so it's driving an inflammatory process and an immune function process to be able to not only deal with those infections, but also to deal with all the other aspects of the disease process with MS. So we were actually very excited by the findings at the biomarker level as well, in addition to the reduction in infections. The FAMS, the Functional Assessment of Multiple Sclerosis, that's pretty much considered the gold standard of functional assessment within MS, kind of like the ADAS-Cog is for assessing cognition and dementia. So you can see that most of the scales were statistically significant. Only the family social well-being and the emotional well-being were not, but otherwise very, very strong, significant findings related to mobility, symptoms, general contentment, thinking and fatigue, and then the total score and the additional concerns. So again, very exciting here that with utilizing the gold standard of assessment of people within or with MS, that we actually showed such significant findings. Very, very, very nice findings. The EQ5D3L is a general measure of quality of life. The Beck Depression Inventory, obviously depression, it's looking at different mood symptoms related to depression. And then the Health Conditions Discomfort Scale, that's again a very broad thing related to different types of conditions that people with MS feel. And so once again, you can see with all 4 of these different assessments, statistically significant as you go through from baseline to 12 months, you get improvements in all of these different scales. So once again, overall, very, very important, significant findings in terms of what the polysaccharides did for people with MS. So overall, when you think about the use of these allopolysaccharides in the context of people with both of these different diseases, they're definitely regulating the cytokines, the interleukins, the growth factors with both of these diseases. We're showing lots of different effects there in terms of what's going on over that 12-month period. Again, with Alzheimer's, you had this really nice clinically and statistically significant effect or improvement in cognition, whereas basically if you look at the 5 FDA-approved drugs for dementia, they do nothing. We reduce the infections in people with MS, which of course is, as I mentioned, the leading cause of mortality in this population. So that's a huge, huge thing, a huge benefit for these folks. improving their overall quality of life and their functional status. That's also very important. As they go through life with MS, they get weaker, they get more debilitated. So to be able to turn that around and help people feel more functional is so crucial. To our knowledge, the allopolysaccharides have no known adverse effects, and they do not negatively interact with any medications or any other dietary supplements. In fact, that may be even actually the opposite. The allopolysaccharides, because of the way they work within our cells, the way our cells need these polysaccharides, they actually make anything else that people take, whether it's medications or other dietary supplements, more effective. And the polysaccharides from aloe actually are so bioavailable that they actually, excuse me, they actually help other compounds, other nutrients, other elements be more bioavailable or absorbable as well. So there's so many benefits to these polysaccharides. And one more point to make is that they're not hyporesponsive, meaning that you don't initially hit some sort of a threshold or a tolerance to these beautiful nutrients. You can take them and take them for life. So it's really, really exciting what we found in both of these studies. Really exciting. Next, we'll talk about the hydrolyzed rice bran, and that's the other side of my polysaccharide story, and how my career has been affected or changed so dramatically. So again, Barbara Kimley introduced me to this particular form of rice bran called hydrolyzed rice bran, and we ended up running 3 different clinical trials in people with no known infection or chronic disease, again, healthy adults, if you will, people with HIV, and then people with non-alcoholic fatty liver disease. So once again, much like with the allopolysaccharides, we showed really, really nice findings, and I'll get into those now. So I mentioned initially, this notion of where polysaccharides come from and the importance of looking at different types of saccharides as opposed to saying, well, sugars are good for you, and then you might laugh me out of the room. But again, it depends on the type of sugar. And so these polysaccharides, again, I've mentioned mannose and galactose, glucose. Again, it depends on the saccharide. It depends on the source of the saccharide. But what we do know about the rice bran, the hydrolyzed rice bran that I'm referring to in particular, is taking rice bran and then hydrolyzing it, or going through hydrolysis with the enzymes from shiitake mushroom. And so this particular compound is actually not even completely characterized. It's such a complex molecule. It's such a complex structure that it cannot even be completely characterized by drawing. So what you're seeing here is just kind of a representation. But what we do know is that when the hydrolyzed when the hydrolyzed result occurs, the molecule is now of a lower molecular weight. And much like I was saying earlier about, are the allopolysaccharides signaling mechanisms for the immune system, I would say the same thing is true for the hydrolyzed rice bran. This is absolutely another signaling mechanism for the immune system. That's a very good thing, not anything negative at all. So again, we ran this first study in healthy adults. We wanted to basically just show, if possible, the effect on natural killer cells. That was really our primary interest in this study. We wanted to see if, based on some of the other research that the company had supported in previous years, if the same would be true. And so we wanted to start out kind of as a first step. Not exactly, I wouldn't describe it as a safety study, although we did look at liver and kidney function markers just to be safe. But we definitely wanted to look at not just the NK cell killer activity or cytotoxicity, but also a host of cytokines and growth factors, much like we did in the Alzheimer's and MS studies as well. We looked at, in this case, 2 different groups. One group took 1 gram of the hydrolyzed rice bran per day, and the other group took 3 grams. So there had been other, work previous to ours where different studies had utilized different treatment protocols. And then this was a very short-term study. This was only for 60 days. So we collected data at baseline, 48 hours, 1 week, 30 days, and then 60 days. And we published 2 papers from this study. The one I'm going to talk about really relates to all of our data. The other one was more of a technical paper that the lab had done some nice, some new novel techniques, and so they published they wanted to publish basically a methodology paper, which was sort of a secondary benefit from running this clinical trial. So here's the graph showing the cytotoxicity, looking at K562 cells, which are typically used to determine how cytotoxic natural killer cells are. This is an in vitro analysis part of the study. But I kid you not, when we looked at these data initially, my colleagues and I, we could not believe it. We literally thought, wow, you could not have demonstrated immunomodulation any more perfectly if you had drawn the graph yourself. So what this shows in the blue line is the 1 gram per day group. The red line is the 3 gram per day group. There's no statistically difference between those time points between the two groups. So essentially you could collapse this into one line if you wanted to. But basically what it shows, and remember, this is in healthy people. This isn't people with heart disease, cancer, dementia, diabetes. This is healthy people. So at baseline, you have your baseline value. It goes up just a little bit at 48 hours, not significantly. At 1 week, it's statistically increased, again, compared to those other first 2 points. But guess what? These are healthy people. There's nothing there. There's no cancer. There's no diabetes. There's no heart disease. And the immune system says, okay, natural killer cells, we don't need you. Shut off. And then it goes back down at 30 days and then is roughly at the same value at 60. So at the 1 week time point, it's statistically higher than every other time point, but it's just a beautiful curve in terms of saying, take this hydrolyzed rice bran, activate, look for something, nothing is there, shut it off. That's exactly the sequence of events. And we could not have drawn this curve any more perfectly to show true immunomodulation according to the natural killer cell cytotoxicity based on treatment with hydrolyzed rice bran. Just such cool findings. The other things, again, everything turned out to be non-statistically different between the two groups. So basically, whether you were taking 1 gram per day or 3 grams per day, it was really of no consequence. And again, we were using, I believe we had 20 subjects in the study, 10 in each group. But TNF alpha and IL-6, 2 classic markers of inflammation, both going down. VEGF, again, a classic marker for cancer and cardiovascular disease, both also going down. And then MCP1 going up, actually, which is very exciting as well. It's going in the opposite direction of the other 3, and that's one of your markers for surveying for cancer. So again, telling the body exactly what it's doing. This is classic food as medicine. This could not be any more food as medicine than this. This is taking that hydrolyzed rice bran, activating the immune system, and looking for things that should not be there. So reducing inflammation, lowering your risk of cardiovascular disease and cancer, at the same time increasing your cancer surveillance. The next study was within people with non-alcoholic fatty liver disease. It was also a short-term trial. This was for 90 days, but we looked at a whole host of markers here. And I don't know if you know anyone with non-alcoholic fatty liver disease, but this is becoming a huge problem worldwide. So along with all of the people that are now overweight or obese, you also have one of the primary effects of that is you have lots of people with fatty liver. Now, you don't necessarily have to be overweight or obese. I think about 10 or 15% of people who are actually normal weight still have a fatty liver. And that is also due to, obviously, poor diet, not exercising, not taking care of yourself in general. So fatty liver is becoming a huge problem worldwide. And much like the problems with dementia and MS, where conventional medicine has a real difficult time helping people with those conditions, same thing with fatty liver. Basically, what people are told to do is lose weight. Well, guess what? Most weight loss programs fail miserably. So we wanted to see if the hydrolyzed rice bran, without modifying diet, without modifying exercise, we didn't do anything else to anyone's behavior. For these 90 days, we simply said, here, take this hydrolyzed rice bran. If they were enrolled in that group, randomly, which we had one group that was taking 1 gram per day of the hydrolyzed rice bran. And then we did do, in this case, a placebo control at many people consider to be a higher standard of clinical trial. So we did have the placebo-controlled, double-blinded placebo control in this particular study. But we looked at people, again, for a short period of time, baseline 45 days and 90 days. And then we looked at all these different biomarkers to see what effect the hydrolyzed rice bran would have on total immune function, the liver enzymes, lipids, oxidative stress markers, and the cytokines and growth factors. So ALP is one of those liver enzyme tests that I mentioned. And you can see the hydrolyzed rice bran group is in blue, whereas the placebo group is in red. Very nice curve here going down to the 90 days. That was a statistically significant finding. ALP is very much linked to changes in liver function over time. So definitely, if you're having that go up, then that's a problem. Eosinophils percentage, same thing here, going up over time in the hydrolyzed rice bran group in blue, whereas it's going down in the placebo group. So this is very important. Again, this is showing that the hydrolyzed rice bran is actually activating the eosinophils in our immune system. Next, the monocytes, another aspect of our immune system, going up in the hydrolyzed rice bran group. It goes up a little bit in the placebo group and then basically plateaus. But again, we're talking about another statistically significant finding in the hydrolyzed rice bran group. And then the neutrophils, along with IL-17, are actually going down in conjunction with each other. And that's a very significant finding because IL-17, actually, if it's dysregulated, if it's out of control, that means you have potentially over a long time a greater chance of having fibrosis in the liver. And we know that neutrophils secrete IL-17. So the fact that in that short-term window of 90 days, the neutrophils were going down, the IL-17 was going down, that's actually a very good finding. And we also, we showed something consistent with previous animal studies utilizing the hydrolyzed rice bran, where they looked at a liver damage model that is similar to hepatitis or fatty liver disease, showing basically the same thing in those studies, that IL-17 came down in response to giving those animals the hydrolyzed rice bran. Gamma-glutamyl transferase, that's one of the most significant clinical markers of liver function. And as that gets worse over time, you're definitely set up for not only fibrosis of the liver, but also cancer of the liver. So this is very, very important to keep that under control. Much like many biomarkers where you don't know where the clinical significance really lies with a particular marker, in this case with GGT or gamma-glutamyl transferase, we know exactly where that value is, and that's at 50. You want to keep that value below 50 so that way you know that your liver is functioning properly. Look what happened here in the hydrolyzed rice bran group. It's a nice curve going all the way down below that 50 mark, whereas in the placebo group it continues going up. So that was a really beautiful finding of this study, and we were very happy with that result. IL-18, again, overall is going down in the hydrolyzed rice bran group, although there was a bit of a dip back up at the 90-day mark, but from baseline it went down, where it's kind of the inverse effect in the in the placebo group, where it peaks at the 45 days and then goes back down at 90. But this is also consistent with the previous animal studies where they showed an overall lowering of IL-18, and that's also very important, lowering that level of inflammation in people with this type of liver disease. So across the board, very, very nice findings in this study. And then finally in the HIV study, we wanted again to look at the effect of hydrolyzed rice bran on a couple of key components of the immune system. This was a little bit longer study. This was for 6 months. Again, looking at people with HIV for a long period of time who were on stable antiretroviral therapy. We gave them a bigger dose. This was for 3 grams per day versus placebo. We had quite a bit more subjects in the study as well. We had 47 people participate. But the issue with HIV today is that it has now become, like so many others, a chronic condition. And one of the things that happens, whereas, very similar to people with diabetes or hypertension or lipid problems, is that people can live for a very long time with these conditions, much like they can now with HIV. It's no longer the death sentence that it was in the 80s and 90s. So the problem is, as people are taking these antiretroviral medications, they're subjected to more side effects due to these toxic medications. So they're at much greater risk. The longer they take these medications, they're at greater risk for things like diabetes and heart disease. So it's very important to monitor what's going on with their medications. And one of the things that happens is that their CD8 cells, their cytotoxic cells, get overstimulated in comparison or in relation to their CD4 helper cells. So once that ratio gets out of balance, it becomes very problematic for people. And then many times the treating physician will try to change or modulate the medication regimen, but 80% of the time that fails. So it's a real significant problem to keep those CD8 cells not from being overstimulated. So we looked at the effect of the hydrolyzed risperidone over a 6-month period. In the CD4, looking at CD4, there's basically no change in either group. Now, even though that's not statistically different, you could argue that on the plus side, CD4s were maintained, or they're stable. And again, these are the helper cells, not the cytotoxic cells. So it's important. Classically, people with HIV, they have a declining CD4 count. And so at minimum, at least hydrolyzed risperidone was able to keep that level, relatively stable. So there's, I guess you could say that's a glass half full looking approach to looking at these particular data. However, on the other hand, we have something really nice here going on, and that is that the CD8 cells are actually coming down according to, giving in response to taking the hydrolyzed risperidone, whereas in the placebo group, they continue going up. So this was definitely a statistically significant finding. So this is really where, we were focusing on in terms of trying to get a handle or a grip on this CD8 overstimulation story. And then much like the GGT value in the liver disease study, same thing is true here in HIV. So if you can keep if you can keep the CD4 to CD8 ratio above 1.0, this is really what your goal is. And as I mentioned, a lot of times the treating physicians for HIV patients are trying to rotate or change their medications to get that value up over 1.0, and 80% of the time that fails. We actually showed here for the very first time, look at what's going on here in blue. The folks in blue, the hydrolyzed risperidone group, actually get above 1.0 their CD4 to CD8 ratio at the 6-month period, whereas the placebo group continues going down. You can see that they were pretty much even at baseline, and then they actually both dropped at 3 months, but then in 6 months, the hydrolyzed risperidone group is above 1.0. So again, just a beautiful finding here. Probably for the first time in literature, anything has actually, from a nutritional perspective, gotten this CD4 to CD8 ratio above 1.0 in people with HIV. And you may remember in the people with Alzheimer's disease, their CD4 to CD8 ratio was actually quite a bit higher. But again, HIV affects the immune system much differently than dementia does. So the fact that we could get the CD4 to CD8 ratio above 1.0 in response to the hydrolyzed risperidone was just so impressive and such a rewarding finding. In conclusion, it's really the same story as the allopolysaccharide in terms of the hydrolyzed risperidone. Obviously, there are a few interesting differences. In particular, with risperidone, we looked at the NK cell cytotoxicity, which is just very impressive. Again, true immunomodulation. We also determined that to be an anti-inflammatory net effect based on what was going on with those cytokines and growth factors. So that was just, really nice, really nice summary of work in that first study. And then looking at people with liver disease, fatty liver disease, and HIV, we showed lots of different findings there in terms of the beneficial effect on various biomarkers in both of these groups, particularly for the folks with HIV, the CD4 to CD8 ratio, and lowering that CD8 overstimulation, really beneficial for people with HIV, again, to try to lower the risk. Ultimately, what we're talking about here is lowering the risk of things like diabetes, heart disease, and cancer in people with HIV. The hydrolyzed rice bran, much like the aloe, has no known adverse effect. Also does not demonstrate any negative interactions with medications or other supplements. And again, it has no hypo-responsiveness. So you can take all of these polysaccharides basically for life and never have to deal with finally hitting a tolerance point where they're no longer beneficial. Again, this is nutrition. This isn't medication. Whereas these polysaccharides are enabling our cellular physiology, medications alter our cellular physiology. So this is a very, very different model from anything that you might think of in terms of the pharmaceutical world. Again, we think not only does the hydrolyzed rice bran, but that the allopolysaccharides both are capable of demonstrating true immunomodulation, which is so important to keeping our immune system functional the way it was meant to be. Just to sum up, I think for me, the most important takeaway from all of this is that really dietary supplements are essential. You cannot get these polysaccharides in the concentrated amounts you need, particularly if you have a very serious health challenge just from food alone. It's really no question in my mind after seeing so many amazing changes in people, not just from the clinical trials that we've run, but from all of the other anecdotal reports that we've gained over just nearly 2 decades now in my work, and going back even in many more years than that with Dr. McDaniel. But these polysaccharides are so important for health, and so I would encourage everyone to make sure that they are a daily part of your life, because again, you're not getting them in your food, and you may be thinking you're eating a very optimal diet, but unfortunately, due to everything going on in the world today with all the pollution, the way food's been changed, the over-processing of our food, you absolutely cannot get these polysaccharides in the diet today, and you really need them from a high-quality dietary supplement source. Thank you so much for your attention. It was my pleasure to present this information to you today. My contact information is here at the bottom of the slide. I would be happy to answer any questions you have. All the best to you and your health.